Hi everyone, my name is Kelly. I'm a PhD candidate in Naguib Meshwar's lab, and I'm really excited to tell you about a project that was in collaboration with the Bazinet lab, Richard Bazinet's lab at U of T, called A History of Child Abuse Associates with Phosphatidylcholine Dysregulation in the Anterior Cingulate Cortex of Depressed Suicides. So first, a little bit of background. We know that up to 32% of Canadians have experienced some form of child abuse, and having experienced child abuse has been shown to predispose individuals to a wide array of different psychopathologies, including MDD and suicide. And unfortunately, over 800,000 individuals die by way of suicide every year. And strikingly, child abuse comprises 54% of the population attributable risk for depression and 67% of that for suicide. And importantly, child abuse is the primary preventable risk factor for the development of mental illness, which is why it's such an important area of research to study. So there's converging evidence in both animal and human studies that suggest that one of the biological vulnerabilities that might underlie this child abuse susceptibility to mental illness is myelination. So the idea is that childhood is a very sensitive period for myelination, and that having the biological cascade that follows experiencing severe child abuse alters the trajectory of myelination and developmental and adaptive myelination. Our group actually has previously reported that in the anterior cingulate cortex, there is an impairment of the myelin program of both the transcriptomic and epigenomics, and that the myelin sheath around small diameter axons is significantly thinner in postmortem brain samples from depressed suicides with a history of child abuse compared to matched samples from depressed suicides and non-psychiatric controls with no history of child abuse. So why might we be seeing these findings? What's the mechanism? One potential area that we were interested in is lipids, because myelin is highly enriched in lipids, with 70 to 85% of the myelin membrane being comprised of lipids, while a typical membrane is about 50% lipids, 50% proteins. And we know that the composition of the phospholipids that make up myelin, so meaning the fatty acid composition specifically, is associated with myelin stability and integrity. So just as a reminder, phospholipids are polar lipids with a glycerol backbone and a phosphate head group with two fatty acids esterified to the SN1 and the SN2 positions over here. So therefore, to investigate this possible relationship between the myelin findings we have before and the lipid composition of myelin, we decided to compare the fatty acid profile of choline glycerophospholipids in the ACC white matter with both concentration and relative percentage metrics, that I'll get to in a bit, between depressed suicides with a history of severe child abuse compared to matched depressed suicides without a history of child abuse and healthy controls. So of the lipid pool of myelin, approximately 40% is comprised of phospholipids, of which nearly 1/3 is represented by the choline glycerophospholipid pool, and the majority of that is phosphatidylcholine, which is why this project is titled phosphatidylcholine. And phosphatidylcholine is a key structural and functional phospholipid of myelin, and is a precursor for a number of different cell signaling molecules, which is why it's interesting to look at. So this is a schematic of the methods. We took a block of anterior cingulate cortex white matter, frozen, usually around 30 milligrams. We extracted the lipids from this block using the Fulch method, which is a biphasic mixture of chloroform and methanol. Then we took those extracted lipids and ran them on a thin layer chromatography plate and isolated the phosphatidylcholine fraction by scraping it off of the silica. Then we took that fraction and methylated it with 14% BF3 methanol. Then these methylated fatty acids were run through gas chromatography, and we were able to get the concentration of each fatty acid in each sample by looking at the area under each peak, which represents an individual fatty acid, and then compare it to a known amount of an internal standard that we added. And based on that, we were then able to gather relative percentage and do statistical analysis. So just as an example, this is what a TLC plate really looks like. And here, I don't know if you can see my mouse, but over here is the choline glycerophospholipid fraction. And this is an example of peaks that we get. So this, for example, is my experimental sample, and this is a standard that contains all of the fatty acids of interest. And then based on the retention time, we're able to match which fatty acids we have, and then go from there. So this is my subject information. All of the samples were obtained from the Douglas Bell Canada Brain Bank, which has a wonderfully well-documented over, I think, close to 3,000 brains. And the phenotypic information is gathered using the validated psychological autopsy method. We also have toxicology data. And so therefore, this well-characterized data allowed us to have at least a decent estimation of the severity of child abuse using validated scales that were administered to next of kin or well-informed informants that were able to gauge the severity along with hospital records and social service records. So it's a combined approach that gets validated information. So since our data did not meet assumptions for regular ANCOVA, we used a bootstrapping approach, bootstrapping resamples the original data set with replacement many thousands of times to create simulated data sets. And this is used to create a sampling distribution. And we know that bootstrap sampling distributions usually approximate correct sampling distributions under most conditions. So here we used a bootstrap linear model with 10,000 simulated data sets. We corrected for multiple comparisons with the Benjamini-Hochberg method. And you can see here that our factors are group, age, group by age, postmortem interval, sex, and pH. We had an age range that from 15 to 85 years old, and we had 101 subjects. So first I'm going to show you the concentration metric. So here what you're looking at is a graph of estimated marginal means, where the black dot is the estimated marginal mean for group, and the purple bars are confidence intervals. So you can see here that the ones in bold are the ones with significant p-values after correction for multiple comparisons. Here is the top bar is control, then depressed suicide, child abuse, and then depressed suicide. So the ones that ended up being significant are C18-2N6, C23N6, and C24N6. And in each of these, the middle bar, so the depressed suicide, child abuse, was the highest. And there was also a trend towards more fatty acids total in this group, but it did not survive correction after significance after correction. So these were the ones that were deemed significant. Then we went ahead and looked at relative percentage, and we didn't actually find any significance. We, although the fatty acids that were significant in the concentration metric had this similar trend, same trend in the relative percentage metric. And the reason why we believe we don't see significance here is because relative percentage does not control for the size of the fatty acid pool. So if, for example, the depressed suicide child abuse group has more fatty acid overall, that's not taken into account here in relative percentage. So this goes to show the importance of having an internal standard. So in this case, we used C17-0, and we're able to get a estimation of concentration in micrograms per gram. So you may have noticed that the fatty acids that we found to be significant in concentration are in the arachidonic acid synthesis pathway. So we aimed to assess whether this finding was reflected in respective gene expression profiles. So arachidonic acid is a precursor to key inflammatory mediators like prostaglandins and leukotrienes. We obtained a list of the genes in the arachidonic acid metabolism pathway from gene ontology. And then we looked for the overlap or the intersection of those genes with existing ACC RNA sequencing genes with a nominal p-value of less than 0.05. So you can see here that there are key genes from the arachidonic acid synthesis pathway like PLA2G4A and PTGS2, which is also known as COX2. They are significantly different between the depressed suicide group and control group. So the purple here is decreased in depressed suicide compared to control, and the green is increased in depressed suicide compared to control. And it's very important to note that this existing ACC RNA-seq data from the same subjects as the lipidomics were from gray matter, not from white matter. So that is quite a significant limitation, but overall, this gene expression differences seems to support a dysregulation in arachidonic acid synthesis or metabolism. But you may be thinking that the directionality doesn't really make sense, because here, these genes, these key genes are, since they're associated with arachidonic acid metabolism into these eicosanoids, you would think that having more arachidonic acid, like in the depressed suicide child abuse group, you would have more of this gene expression. So it does seem that the findings might be contradictory, but we think that there's several reasons that might be able to explain this. So firstly, the gray versus white matter. Also, we might be seeing some compensation, some compensatory mechanisms. And there's also a lot of regulation of this pathway. And for example, we know that a really well-documented biological consequence of child abuse or early life adversity is a dysregulation of glucocorticoid system, which could potentially inhibit this step. So there are a lot of different reasons why this directionality might be observed, but we don't know. This is kind of all speculation right now, but I think what we could conclude is that when you combine the genetics and when you combine the fatty acid quantification, there seems to be a dysregulation in the arachidonic acid metabolism and synthesis in the depressed suicide child abuse group. So then we went ahead and we looked at the different covariates. And you can see here in this correlation plot, especially in the concentration, that age is the most highly correlated variable with the fatty acid metrics. And since we had such a wide range of ages, we were able to take advantage of that and do Pearson correlations to look at the relationship between age and fatty acid concentration and relative percentage. So you can see here that most of the fatty acids tend to decrease with age, with some slopes being steeper than others. I'm showing only the significant p-values here. And when it comes to relative percentage, what's interesting is that all the fatty acids that were not decreasing with age are represented here in the relative percentage. So it's basically just saying that it composes a higher percentage of the total because it's not decreasing with age. And the ones that we see here that are decreasing with age were the ones with the steepest slope here in concentration. So again, the concentration metric does seem to be important in order to gain the most collectively accurate information. And here I just have a summary table explaining what we see. So the study is not without limitations. It was underpowered, so you're not able to detect significant differences between individual pairs of groups in post hoc tests. So we can only say that there is dysregulation between the groups. We also only examined the choline glycerol phospholipid pool of phospholipids, so it was not possible to get a complete picture of any global dysregulation without studying other lipid classes, which is why I have an ongoing project now that's going to look at all of the phospholipids plus cholesterol to get a more comprehensive picture. We also had a low number of females in our sample, so we were likely unable to reliably detect sex-specific differences, though I did see a difference in C18, 2, and 6 fatty acid in sex, but we would need to validate that with more samples. As mentioned, the RNA-seq data is from gray matter, while the FA quantifications are from white matter. And the available RNA-seq data only compared the depressed suicide, child abuse, and control samples. So the differentially expressed genes could not be specifically associated with child abuse without further validation from the depressed suicide group. And lastly, we do not know the source of these fatty acids, so we can't comment on whether the differences that we see are due to alterations in synthesis, in transport, or dietary intake. So we don't exactly know where these differences come from. We can just see that they're there. So in summary, what we can say is that the concentration of several key fatty acids in the arachidonic acid synthesis pathway are dysregulated between our groups of interest. And this was further evidenced through the gene expression. The concentration of most fatty acids, as well as the total fatty acid concentration, tends to decrease with age. And the concentration metric, which uses an internal standard, can be useful to prevent overlooking or mischaracterizing findings that only consider relative percentage due to the consideration of the size of the total pool. So if you're interested in learning more, you can read these results, which were published in Translational Psychiatry. And I would like to thank everyone for their attention, and also my supervisor, Naguib Meshwar, and the PI at the University of Toronto, Dr. Richard Basnett, as well as the entire McGill Group for Suicide Studies. So thank you, and I'd be really happy to take questions.
Thanks, Kelly. Really nice and clear presentation. So questions for Kelly?
I think there's one from Nico, yeah.
Yeah, just a quick question. Very, very interesting presentation, Kelly. Congratulations for this nice project. A quick question about your what you call the healthy controls.
Yes.
Do you have, by chance, any information about possible child abuse in these subjects?
Yeah, so we did the same psychological autopsy on all the subjects in the brain bank, and that includes the child abuse scales and investigation, and any subjects with either confirmed or possible history of child abuse, any indication of child abuse, they were automatically excluded. So that goes for the healthy controls as well as the depressed suicides without child abuse. And also the child abuse subjects that were included had to have the highest severity of child abuse as measured by the CECA scale. So that's how we kind of made those differentiations.